This method can help answer key questions in the microbiology field, such as how fluid flow alters fungal biofilm growth and development. The main advantage of this technique is that it allows us to quantitatively evaluate the effects of flow on biofilm growth and development in real time.
Begin this experiment with assembly of the flow apparatus as described in the text protocol. The day of the experiment, attach the bubble trap, temperature probe, and all single-use components. To assemble the filter, remove the plunger from a 1 milliliter syringe to make it an adapter. Force the tubing from the filter bottle into this end, and attach a 0.2 micron filter to the tubing leading to the growth flask. Apply silicone vacuum grease around the barb of the slide adapter prior to connecting it, as this helps prevent air leaks into the system. Fill the attachment flask with 100 milliliters of YPD, and fill the growth flask with 200 milliliters of YPD. Ensure that the green side tubing reaches the media in each flask. Ensure that all valves are open. Attach the bubble trap to a vacuum and connect the pump to the green side tubing downstream of the bubble trap. Pump the fluid at a flow rate of 3.3 milliliters per minute to completely fill the green side. Then dispense and discard approximately 1 to 2 milliliters of the media, because the first couple of milliliters often contain dead cells or random debris. Ensure that the green side of the tubing is filled with media and has no bubbles downstream of the bubble trap before proceeding. Fill the channel slide and the reservoir with YPD, taking care not to introduce bubbles. Connect the slide to the flow apparatus.